Thank you for inviting me to BioHabana. I'm so excited to be here today to share with you my talk on molecular mimicry through the lens of the microbiome. I'll be covering microbial and dietary autoimmune triggers in Parkinson's disease. I'm Martha Carlin, CEO and founder of the BioCollective. My journey into scientific research began in 2002 when my 44-year-old husband was diagnosed with Parkinson's disease. And I decided to take my business background and apply it to the science of studying Parkinson's. Since that time, I've studied many different fields of science. I'm known for my out-of-the-box thinking and systems-based approach to tackling the problem. In 2015, I founded the BioCollective, and we started to collect fecal samples across the population, but with a focus in Parkinson's disease in order to connect the dots across the population to what is happening in our microbiome. Along the way, I interviewed many people with Parkinson's, looking for patterns in the patients that I talked to. And some of those early clues actually point to molecular mimicry from gut dysbiosis. But early observations in my interviews were many childhood infections, streptococcus in particular, multiple courses of antibiotics, often antibiotics in the teens for acne treatment, the smell of ammonia, herpes cold sores, and shingles, just to name a few. I also had the opportunity early in my career to work with a large hospital data set of 10,000 Parkinson's patients, and we were able to look for comorbidity data where the most significant comorbidities were in insulin resistance, diabetes, and dyslipidemia, cardiovascular health. And then our fecal sample collection, of course, I'm going to talk a bit more about that. Early on in the process, I came across this paper from Dr. Isaac Ginsberg, the role of bacteriolysis in the pathophysiology of inflammation, infection, and post-infectious sequela. Dr. Ginsberg is a prolific researcher who was still publishing well into his mid-90s when he passed away last year. And when I found this paper, I saw many correlations to what I could see in the history of people with Parkinson's and in my husband's own history. And I've highlighted here in red some of the key things that we've actually found through the research in our microbiome data. And Parkinson's is, at least in part, an infection that goes on to produce long-term oxidative stress, moving to hypercoagulation, amyloid formation, disruption of the innate immune system, and systemic inflammation, particularly in the brain. There are a number of circulating inflammatory biomarkers that have been identified. Iron overload as well, identified in certain parts of the brain, and more recently circulating bacterial inflammagens, LPS, and lipoteichoic acid. These going on to develop into alpha-synuclein aggregation, mitochondrial dysfunction, and neurodegeneration. I'll talk a bit about some of the biomarkers that we have identified that overlay with this existing research. This slide is densely packed, but essential for explaining the pathogenesis of post-infectious sequela as outlined in Dr. Ginsberg's 2002 paper. The interaction between bacteria, whether gram-positive or gram-negative, and cationic peptides from neutrophils or beta-lactam antibiotics. activates autolytic wall enzymes such as muramidases in bacteria. This activation can be inhibited by polysulfates like heparin or by antibacterial lytic antibiotics, though these are not depicted on this slide. This activates the M1 macrophages. Components like lipopolysaccharide, lipoteichoic acid, and peptidoglycan are released following bacterial lysis and can interact with macrophage surfaces via CD14, toll-like receptors, or phospholipid binding sites. Endocytosis of the bacteria, or peptidoglycan, may induce granulomatous inflammation due to the inability to degrade the wall components. Or it may also trigger cell activation, resulting in generation and exocytosis of oxidants, cytotoxic cytokines, hydrolases, phospholipases, and antimicrobial peptides. LTA and PPG can activate complement and coagulation cascades, leading to disseminated intravascular coagulopathy. Ultimately, that results in tissue damage and can result in chronic inflammation. There is evidence of M1 pro-inflammatory activation in the Parkinson's brain, suggesting that the underlying pathology subverts normal remediation of pro-inflammatory pathways for conversion to M2, which are involved in scavenging and healing. This key point in Dr. Ginsberg's discussion of post-infectious sequelae highlights that while M1 macrophages are activated, the subsequent M2 scavenging and healing macrophage function is disrupted, leading to chronic inflammatory cycle where the healing pathway is impaired. So how might microbes contribute to Parkinson's pathology? Many bacteria produce pore-forming toxins that create pores in the gut lining. This porosity allows the release of light chain amyloid, which catalyzes blockage of neurotransmitter release and produces heat shock proteins. Under stress, bacteria and human cells release small peptides known as heat shock proteins. These toxins, released by bacteria as they die from antibiotics or immune system attacks, are not always fully cleared from the system. And you can see here just a small listing of literally hundreds of bacterial products and protein toxins. And many of these can slow the speed of transit, and constipation leads to toxin buildup. Constipation is actually a prodromal symptom in Parkinson's that can precede a diagnosis by as many as 10 to 15 years. So what is molecular mimicry? Recognition of commensal bacterial epitopes by T cell receptors. So the bacteria produce a heat shock protein with a similar sequence to a human tissue, and the microbial antigen that is produced will migrate to the tissue where the cognate self-antigen is expressed and elicit an autoimmune response. So, for example, you might get a self-antigen in the pancreas, which would be correlated with diabetes. And there's quite a bit of evidence connecting autoimmune disease to diabetes, uveitis, MS, and lupus. I've actually added Parkinson's here because I think that evidence is growing. So what are heat shock proteins? They're proteins produced by cells exposed to stress. They're found in virtually all living organisms, from bacteria to humans. They're called heat shock proteins, but they are produced in response to all kinds of stress, not just heat: nutrient stress, oxidative and nitrosative stress, UV light, temperature, wound healing, tissue remodeling, just to name a few. Many are chaperones, stabilizing proteins to ensure correct folding, alpha B crystalline being one of those most important to ensuring correct folding. This is a diagram of our early evidence suggesting that heat shock proteins from bacteria and food may contribute to autoimmune triggers impacting Parkinson's. Greater exposure to antibiotics, changes in diet, herbicides and pesticides can alter the microbiome. And this greater exposure leads to less clearance of the pathogens, a more pathogenic microbiome, increased production of heat shock proteins such as alpha crystalline, heat shock protein 65, and cardiolipin. The induction of antibodies and T cells to human alpha crystalline and cardiolipin. Antibodies then neutralize the human alpha B crystalline, heat shock protein 60, and human cardiolipin via molecular mimicry, leading to misfolded proteins and alpha-synuclein aggregation. Our research has also indicated an increase in polyamine production and specific polyamines leading to increased aggregation. And research has also shown that these aggregated alpha-synuclein are actually moving via the vagus nerve to the brain. So this is some of our preliminary data working with Dr. Paredes at the University of Denver on polyamine and kynurenine content in the fecal samples from our collection. The bottom diagram is the taxa that from our earliest study that were associated with Parkinson's disease. Akkermansia muciniphila can damage the lining, the gut lining, as it produces mucinolytic enzymes. Porphyromonas bananus, a relative of the oral pathogen Porphyromonas gingivalis, has been also associated with Alzheimer's disease and gingipane production. Methanobrevibacter smithii has also been associated in a number of other studies with Parkinson's disease. And Streptococcus, which I mentioned earlier. On the right-hand side is just diagrams from our research with Artigen Inc. in Poland. These are 3 different genes that were identified as markers in our Parkinson's microbiome data that are associated with mycobacteria. And it's interesting because mycobacteria are often missed in microbiome research because they are difficult to lyse and low abundance in the sample. But there is growing evidence that Mycobacterium avium paratuberculosis in the milk supply is associated with a number of other autoimmune diseases, and we are doing some ongoing research with Dr. Thomas Dow, who is well published in that area. So here are some potential autoimmune triggers in Parkinson's. As I mentioned, we've identified markers associated with mycobacteria, which produces an alpha crystalline, a heat shock protein 65, and a cardiolipin. Wheat, which actually has the closest homology to human alpha crystalline. Haemophilus influenza, which is actually one of the top taxa in our work with Artigen, has sialylated LPS and cardiolipin. There are numerous LPS models of Parkinson's disease. Streptococcus, again, continues to come up. Streptolysin O and superantigen A producing microbial antigens to that are similar to cardiac myosin, laminin, and vimentin. Porphyromonas, again, cardiolipin. That is the cardiolipin is the mitochondrial membrane wall. H. pylori is O antigens and Lewis blood group. And then corn, which produces an aquaporin antigen and cry-AB that are also, cry-AB is homologous to the human alpha-B crystalline, and aquaporin 4 is a brain aquaporin. So there are many examples of molecular mimicry, those shared epitopes between humans, food, and bacteria that can lead to dysbiosis, B or T cell reactive epitopes, cross-reactivity at the protein level. And again, that's just those key areas that we've been looking at. Alpha-B crystalline, which manages protein misfolding, heat shock protein 60, which has many actions, heart myosin, myelin glycoprotein, HLA-B27, and the aquaporin that I mentioned as well. So here is some published research from a group out of Poland on the role of heat shock proteins in Parkinson's, where they showed statistically significant differences in both IgM and IgG antibodies against alpha-B crystalline. And this was a time series study over 13 months, showing an increase in the autoantibodies over time. And then here's another publication connecting this potential autoimmune triggers, genetic remodeling of protein glycosylation. And you will see later from some of our data that we have identified genes involved in glycosylation as the top marker in the microbiome data. So heat shock protein 60 and 65, mimicry, it's ubiquitous in human tissues. It's a mitochondrial protein. It can elicit a complex autoimmune response characterized by multiple cross reactions due to the mimicry. It's found in other intracellular sites and extracellularly as well, and in circulation. And the heat shock protein 65 in bacteria, which is homologous, prevents apoptosis of macrophages. Let me go back. And it's used to distinguish species of mycobacteria, and it's highly biologically active. So here, just an example to show you how you can look at these alignments. Across the top, we have the Homo sapien heat shock protein 60. And as you move down here, you can see here we have the plant phosphatidylinositol 3-phosphate with homology. You see the overlap between those sequence areas of the genome. And then at the bottom, the actinobacterial trypsin-like peptidase with the overlap as well. And we have found a higher amount of actinobacteria in the Parkinson's samples as well. And this is going to be kind of tiny and hard to see, but this is where, we look. So the red is the sequence alignment where those shared amino acids are, and a shared epitope, say, due to 20 to 30 shared amino acids highlighted in red sequence, that's an indication of a homologous epitope. So our Parkinson's cohort that we collected samples from and used in our Artigen machine learning data was 58 Parkinson's patients and 48 healthy controls. The stools were collected using the biocollector kit, collected the whole stool that was processed and aliquoted, and then we did whole genome sequencing, and that data was provided to Artigen Inc. in Poland, a company that does biomarker identification in cancer primarily, but we brought them along our Parkinson's journey. And here, the most important genes for Parkinson's prediction from the Artigen model were this WBGU gene involved in glycosylation, which is homologous to the human insulin GALE gene, and this NSRR gene, which is a gene involved or an indicator of nitrosative stress and maybe an indication of the microbiome trying to adjust to and rebalance things. The IOLJ, which is a myoinositol osmoregulator, and GROS, a protein kinetic trap release, which is involved in protein folding. In the taxonomic analysis from our work with Artigen, Haemophilus influenza was by far the strongest marker in the taxonomy. And again, back to our previous discussion, Haemophilus does produce a cardiolipin, an opacity protein A, and sialylated LPS, where we are looking at molecular mimicry with Nu5AC and mitochondrial cardiolipin. So, in conclusion, immune response is a protective process that can go awry. It can get subverted by molecular mimicry, and the immune response targets our homologous human cells. Depending on the molecule, there are different signs and symptoms, and we know that Parkinson's has a heterogeneous symptomology. Molecules arising from this process may be contributing to the pathology of Parkinson's. Osmotic stress is a key contributor to heat shock protein production, and osmotic stress is clearly evident in Parkinson's stools, something we have not published, but we did identify in our sample collection that the people in our lab could identify people with Parkinson's just by looking at their stool. And with that, I will take questions. And thank you again. I so appreciate you having me as a part of your conference.